If nano is cool, and bio is cool, and photonics are cool, then what could possibly be cooler than nanobiophotonics? Let's see how cool nanobiophotonics is. So we talked a bit about nanobiophotonicks could be a lot of different things. It could be shotgun DNA sequencing, which is the technique for which Illumina is most well known. Shotgun DNA sequencing, where we take the fluorescently labeled base pairs and flow them across a DNA microarray that has different sequences, different overlapping sequences at different points on the array. And then if we reconstruct the sequence of which spots light up and which colors where, we can reconstruct the entire genome in a much shorter time than it would take to reconstruct it if we took the DNA individually. But the techniques that I'm going to talk about today rely primarily on plasmonic properties. So plasmonics, plasmons are quantized oscillations of the conduction electrons in a metal. And a surface plasmon gives rise to the color and light confinement effects in metal nanoparticles, and there's a propagating form of a surface plasmon called the surface plasmon polariton, which allows you to waveguide light through photonic circuits if you have a particularly flat metal dielectric interface, like a flat metal film, and you can actually use it as a waveguide in the same way that you could use a dielectric waveguide, say an optical fiber or a polymer waveguide. So plasmons are an effect of the looseness with which the conduction electrons in a metal are held. And not all metals hold their conduction electrons equally well. That's why metals have different work functions and ionization energies and so on. The conduction electrons have a plasma frequency, which is the maximum frequency of a photon that can be reflected back by into the atmosphere or into the exterior environment. And the plasma frequency is what gives rise to the metallic sheen and reflectivity. This is silver. Took these images from Wikipedia. This is silver and platinum, which are silvery, and they reflect all wavelengths in the visible spectrum because their plasma frequency is in the deep UV. So it's a little bit like having a tennis ball and a tennis racket. I haven't focus group tested this analogy, so it's not going to be exactly right. But say you have a tennis ball, and you whack the tennis ball into the chain link fence. And if you do it kind of with not too much energy, the ball will kind of bounce back. If you hit it sufficiently hard, it'll get lodged in the chain link fence, right? And if you hit it so hard, it will actually pass through the chain link fence. And the same sort of thing happens in metals. So if you have a really high energy photon that is more energetic than the plasma frequency, so the frequency of the photon is more energetic, it will actually pass through the metal. But that's usually beyond the visible range, but not always. And what are some examples of metals that have plasma frequencies that are in the visible range? And they could be copper or gold, which allow some light through, and they appear, or they allow some light through, and they reflect what appears to us a reddish or orange or gold hue. At the surface, so the plasma frequency is a bulk phenomenon. But if you shrink down the particles, and this is where we can start to engineer these particles, if you shrink down the size of the particles, you actually change the plasma frequency and you move it toward the visible regime. And what the phenomenon that gives rise to this is called surface plasmon resonance, or if you have a metal sphere or any metallic nanoparticle, but not just any metallic nanoparticle, but any nanoscale feature of metal. It could be a flat film that has some asperities on it that resonate with localized surface plasmons. So surface plasmon resonance is an oscillation of the conduction electrons about a metal dielectric interface. And these resonances occur at peak wavelengths that correspond to the size and the dielectric environment. So if you change the dielectric environment, say from air to water to silica to no protein bound to a protein bound to these particles, you change the dielectric interface and therefore you change the plasmon resonance. Yep.
So a constant E field, say in the up-down direction of this image, would cause that oscillation? It will start springing back and forth and continue to do so?
Yes. In the case where the electric field is in resonance with the localized surface plasmon resonance of these particular particles in this particular interface, these particles will both absorb and scatter light at that frequency. It's almost like a jump rope tied to a wall on one end, and it will resonate at a particular frequency if you put the right input into it.
We need an oscillating field.
You need an oscillating field. Yeah. But an oscillating field is automatic in the case of a photon because a photon is a wave. So if you have just a direct current field, you'll get polarization, but it won't be oscillatory. Okay, and you can have different modes within the same particles. You can have dipole modes where the electrons just slosh back and forth like this, and you can have, that's here, and you can have quadrupole modes where the electrons will go from one side to another. You can have other types of modes for anisotropic particles. These are just spheres like this, but you can have, say, rings and split rings that will resonate in different ways depending on the wavelength and also polarity of the light. Okay, how is a color perceived? We talked about this earlier in the class. Absorption is in a case where an object absorbs, the apparent color is everything but what is absorbed. And in scattering, the apparent color is what's scattered. So in this case, this slide I took from Nano Composites, which is a local company that makes specialized nanoparticles, and their website is actually very useful in the descriptions and the images that they show. So say you take white light, which is a combination of all the colors of the rainbow, and you send it through this material that absorbs with this kind of absorption profile. Say it's strongest here and then it tails off. So it's strongest in the green, it absorbs the green and what's left over, but mostly red, and that's how it appears, this solution appears to us. These are actually gold nanoparticles. Notice they don't look gold because we've shrunk them so far down that the plasma frequency has moved into the visible. Now look at the scattering frequency. So if you take this light that's a combination of these wavelengths, impinges on a material, and the scattered light is actually just the wavelengths that are scattered. So absorption and scattering typically occur at the same wavelengths, but some particles are better at scattering and some are better at absorbing. So if you have two different particles in the same material, you can get dichroic effects where the color of reflection and scattering is different from the color that's due to absorption. And this is the Lycurgus cup. It's like 1600 years old from the Roman Empire. It's a remarkable example of dichroic glass that's actually an early example of unintentional nanoengineering. So they had silver in the glass, and they probably knew that there was silver in the glass, but there was also gold in the glass. And the silver and the gold nanoparticles that formed during the glass making process, one of them scatters more at a particular frequency, and one of them absorbs more at a different frequency. So as a result, the scattered light is dominated by the thing that scatters the green light, and the red upon transmission of light is due to the fact that the other kind of particle in here is absorbing like blue or green light and allowing red light to pass through. So metal nanoparticles can actually be tuned like quantum dots, in fact, to appear certain colors. So these are all metal nanoparticles. I believe these are all silver, but I could be mistaken. But the point is that you can take metal nanoparticles that have different absorption profiles, and you can generate all the colors of the rainbow. They're not fluorescent, as in the case of quantum dots, but they do appear to be these different colors. And you can now engineer this types of effect just by mixing these things together that have different absorption and scattering cross sections. So this is a vial of something that actually has the reverse effect of the Lycurgus effect, because it scatters red and it appears blue-green through absorption. Now, how can you use this technique to sense biomolecules? So this is done ubiquitously in the drug discovery industry. It's a technique just called SPR, for surface plasmon resonance. And what you do is you take a prism or this half dome type shape, and you have these microfluidic channels that have some bound antigen or antibody, and you're trying to detect the binding event. Now, what this binding event from this analyte in the solution does is it changes the local dielectric environment. So it changes the plasmon resonance of this metal film, which is almost always gold. So this is a plot. This is signal versus time, and what happens is that when the plasmon frequency changes here, the intensity of this scattered, of this reflected beam is attenuated, because so you tune the angle so that you get the maximum amount of absorption of the film. So you shine this laser in, and then there's a detector over here. Maybe it's not a laser. You shine this light source in. There's a detector over here, and you find where the minimum in intensity is of the reflected light, and that is where the plasmon resonance is of the gold film. If you change the dielectric environment by having these guys bind to the receptors at the surface that are chemically adsorbed to the surface, then the resonance frequency of the gold changes, and therefore the intensity of this changes. Now, as you go, let me see if I answer the question. As you go versus time, you have no binding here, so we're down here. Then we have binding, and then the change in intensity goes way up. Then if we stop flowing the analyte starts to desorb. Now notice that we're getting kinetic information here too, so we can tell over time how these signals decay, how they're induced, and then how they decay. So we can get the binding kinetics and the strength of binding, because we know the concentration in the solution as well. And then we can learn something about this protein ligand interaction. Yep.
So it's more of a structural question. Looks like a chip we insert into a microfluidic device. But do we see the proteins that we're testing for a ligand, right? And we need to attach the proteins to the gold foil? Mm-hmm. Okay.
Yeah. So before we do this, we've done some chemistry, some called conjugation chemistry, where we conjugate this receptor molecule to the gold film. And these instruments are sold commercially by General Electric, called Biocore or Biocore. And if you go to this YouTube video, you can see this fully animated. And a lot of different companies have, or different groups have done really nice animations of how this works. Okay, so what it allows you to do is label-free detection of analytes. So you don't need a label, but you do need to modify the surface somehow. So for example, if you get, or your older male relative has gotten a prostate-specific antigen test, for example, and there are many, many examples, but they use the SPR technique to measure the presence of this marker, which is a marker for prostate cancer. It can also detect viruses and ions. And the change in the local refractive index reflects the change in reflectance maximum when the ligand binds. So that's basically just what I told you, that the intensity of this reflected beam is changed by the binding of the analyte. So it can tell you, is there binding, yes or no, specificity for this analyte to this given receptor, the affinity, and also the kinetics and the time dependence. So, yeah.
The question on function. So this is due to a change in the local electronic environment. So we see a different photonic response because there's more mass and therefore more area for the electrons to move around in once it binds?
We change, so in answering the question, I would have to get way too technical for my pay grade and also for the purposes of this course. But basically the mass of stuff bound here increases, I don't want to say if it's increased or decreased, the dielectric constant of the medium, probably, no, I'm not going to say because I can't think of it off the top of my head. And the resonant frequency of the gold changes. And we're going to do, we're going to talk about plasmon resonance with chalkboard on Wednesday next week to do this in a little bit more detail. I'm just going from the top down now, so you know why it's interesting before we tell you how it works, as opposed to general chemistry classes where they tell you how everything works and then you don't know why you should give a shit. And concentration information. Surface plasmon polariton is a propagating surface plasmon that is called that's called a polariton. So usually you need a long metal object to generate a polariton, and this is something where, say you have a long wire and you put light into one end, the surface plasmon will actually propagate from one end to the other and then scatter out the other end. And I'll show you some examples of how this is useful. So it can be used to confine light to wavelength dimensions and also nanoscale dimensions for photonic circuits. So you could have a photon of say 800 nanometer light and confine it to like a 200 nanometer line width, and then it would be transmitted at the end, or scattered at the end of the wire. Okay, so I showed this slide last time, and these are some examples from the literature. None of this is going to be on the exam, but thank you for coming anyway. And so the goal is to cross 2 nanowires at 90 degrees. Why would you want to do that? So we got this one, which is close enough. So you can couple photonic and plasmonic waveguides. For example, this is a polymer waveguide. It's just a dielectric material. And you can couple light into this silver nanowire, and it scatters at the end. You can also take a nanowire. This is a silver nanowire and a quantum dot. And what you're doing here is you're exciting an electron hole pair in the quantum dot, and then when the electron goes down from the conduction band to the valence band, it releases its energy. But it doesn't release its energy in the form of an emitted photon. It actually releases its energy directly into the surface plasmon mode of the nanowire. So this is a near field, it's called a near field phenomenon, where the energy is directly transmitted, it's not mediated by a photon. It's just a proximity effect, and the dipole energy here is automatically transferred electrostatically. Then the surface plasma polariton goes to the other end, and then the fluorescent photon is actually emitted here. So the fluorescent photon is not emitted by the quantum dot, but it is emitted here. And there is there's the paper. Now you can do something pretty interesting with this. This is like the Rube Goldberg experiment in plasmonics and nanophotonics. So the idea here, I'm not exactly sure what the technological importance of this was, but it is a really cool demonstration, and it gets cooler as well. So what we want to do is take a plasmonic nanowire, the silver nanowire, and cross it literally with a germanium semiconductor nanowire. Then what we're going to do is we're going to raster a laser across this, this image. So this is an SEM image, and overlaid on this is a rastered image of output, of scattering output due to scanning the light. So maybe it'll make sense once I point to it. Okay, so you start here. And what happens here is you shine light on the tip of the silver nanowire, and some light is scattered, and that's registered as a and some okay. Some light is scattered, but some light also goes into the surface plasmon mode in the nanowire, excites electron-hole pairs in the semiconductor nanowire, and those electron-hole pairs are separated by means of the fact that this nanowire is not bisected exactly in the middle. So the electrons go preferentially one direction, and the holes preferentially go the other direction. And then there's a machine over here, just an electrometer, that measures the current. And the measurement of current is what produces the spot here. It's not the actual scattering event. It's the fact that we measured current when the laser reached this point. Let me get to the end and see if I answer it. Okay, then we keep rastering the laser, nothing, 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 because the smooth sidewalls, you need irregularity in order to produce the scattering event. The smooth sidewalls don't allow you to couple light directly into the nanowire. You need the ends. So you get here, you get here, you get here, then you're exciting the germanium nanowire directly. Then you're measuring the electron hole pairs that go to the electrometer, and you get the current, like negative current here and positive current here, due to the asymmetry in the junction. Then you get to the very end, and again you have light that goes in to the silver nanowire due to surface plasmon polaritons, then excites an electron hole pair here, which is separated, goes into our electrometer, and then we measure a current. So these are overlaid maps. One is the photocurrent as a function of position, and the other one is just a scanning electron micrograph. And what we've done here, we send a photon in here, and we measure the current out here. So that's pretty cool, right? These are nanostructures that are directly transducing this optical energy without being mediated by photons. Yeah.
See if I understood you correctly. You say that we only see the scattering event, or is it we're going to see a current up here at I plasma 2 because we're making the current measurement? So we're dealing with current in the order of picoamps. So I'm wondering if the actual making a measurement provides enough resistance that something in the circuit changes so we see the output, but we wouldn't see it if it wasn't connected? So the question is, this is not, there is scattering here, but this is not what this signal is. This is a photocurrent signal. There is scattering, and there's also coupling of the, you need scattering in order for energy to couple into the nanowire. So there is scattering here. It looks like scattering, but this red spot was not generated by scattering. There's no optical micrograph here at all. This is just an SEM and a photocurrent map. Yeah.
So if I understand correctly, literally the photons from the light source are being transmitted directly into electrical current.
Yes, directly through this quite convoluted mechanism, and it gets worse in the next slide. So I want to make sure that we're okay here. Yeah.
So wouldn't putting a laser through metal cause a photoelectric effect to have current all through the wire? Why is it only mechanical?
Yeah. So that's a good point. The silver nanowire. So it has something to do with the momentum vector mismatch between the incoming photon and the modes and some physics gobbledygook that I'm not going to go into, and I say that because I don't completely understand it. But the fact is you need to have the scattered frequencies in order to find some of those scattered frequencies that match the allowed momentums of photons that produce a surface plasmon polariton. I'm embarrassed that explanation is on YouTube, but there it is. Is that okay for now? Smooth features are much more difficult to couple light into an object. You need roughness in order to generate this plethora of scattering in order to get the photons to couple into the nanowire. So therefore, it only works at the end. Okay, now we're going to sneeze some quantum dots on the whole thing. And why do we want to do that? Because we want to make one more set of complexity. So now this is these are where the quantum dots ended up, and it's kind of this random arrangement of stuff. And so here's a quantum dot, here's a quantum dot, here's a quantum dot, here's a quantum dot, and what happens here is that we irradiate where this quantum dot is, and what we get is photocurrent. So we've irradiated this quantum dot, and the quantum dot is here. We're not shining at the end of the nanowire, or that end of the nanowire, or the germanium nanowire directly. We're shining it here, or we're shining it everywhere, but the system responds when you shine it here. Why should it respond? Why should you get photocurrent when the quantum dot is illuminated? Well, the reason is that the quantum dot has an electron hole pair that's been generated, and then instead of emitting a fluorescent photon, the energy couples directly into the surface plasmon mode of the nanowire, propagates down here, it's separated into the germanium nanowire, and then you register it as photocurrent. So we've added one more of these so-called near-field phenomena that transduces optical energy without the emission of any photons at all. So you shine, the only thing we've done in this little region of the plot is excite the electron hole pair in the quantum dot, and then that energy after this transition and this propagation and this transmission and this propagation, it's finally registered as current, and that's why it shows up as a red spot. That's pretty cool. Okay, when I said that is not on the exam, but surface-enhanced Raman scattering, you should know that surface-enhanced Raman scattering, we already have been through this in the course, but I want to show you some bio applications of it. So basically what happens is that, and this is review, but you should know it for the exam, where you have, or at least just what it is for the exam, and you have this vibrational state, and a photon comes in and changes the vibrational state of the molecule, and then a photon is scattered at some frequency that's either increased or decreased, depending on whether energy was borrowed from the vibrating molecule in a way to reduce its vibrational frequency, or increases, or the frequency. Let me start over. Vibrating molecule, because I'm getting all of my respectives mixed up. The vast majority of the cases, what happens is that the vibrating molecule is vibrating, then the photon comes in, and then we have an inelastic scattering event where the vibrational frequency increases, and then the energy of the scattered photon is reduced by the amount that the vibrational frequency increased. That's called a Stokes shift. Don't worry about that. But the opposite case can also happen, but it's less probable, where we have an anti-Stokes event, where we have a molecule that's already in the excited vibrational state, and the incoming photon knocks it down an energy level to a lower energy level, but that energy goes into the scattered photon, and then you have a higher energy photon than the one that was incoming. Yep.
So does the Raman device account for that? Integrating it just sort of doesn't, irrespective of sign, just looks at the deltas?
Yeah, that's a good question. Usually a molecule is, we're almost always dealing with Stokes shifts. Okay, so here we have in kind of schematic form, we have this excitation light, and it starts here, and then it ends here, and this is the Stokes shift, and then there's a scattered photon, and then we have one that's already in the higher vibrational state, goes up to some virtual state, and then it goes down to a lower energy state. Okay, and a Raman spectrum looks something like this. And this is a molecular fingerprint. This is an experimental apparatus that you might use for surface-enhanced Raman scattering, or SIRS. And you have your cluster of silver nanoparticles that are mixed with some analyte, say. Then you shine in a near IR laser, and it bounces back, and some of the laser passes through this mirror, and then you take the spectrum that exits, and you subtract off the spectrum that you shined in to begin with, and then you get the spectrum. Okay, so this is dopamine, a molecule, and norepinephrine. They have different, even though they're similar chemical structures, they give you completely different Raman spectra. So you get a fingerprint. Don't worry about what all these peaks are. They basically have to do with the vibrations of all of the different bonds in the molecule. Remember, vibrational spectroscopy. So what can you do in biomedical applications? So for example, you can get these cells to endocytose these nanoparticles. So they take them in, and if you have a monolayer, a chemical monolayer on the SIRS active nanoparticles, you can actually detect the pH based on whether or not, say you have a basic or acidic group on the nanoparticle, that can be used as a pH probe. So you take this cell and you put it under a SIRS microscope, and based on the strength of the signal, you can detect what the pH is in different parts of the cell. You can also do this in whole animals. So you take this mouse and you take its, well, it's disgusting, but you make an optical window. Don't ask how that was done. Then you shine light in, and even though the body of a mammal is just full of different stuff that gives you different Raman signals, because the fingerprint of the Raman analyte is so unique, and the surface-enhanced Raman scattering spectrum amplifies it to such an extreme degree, it's actually possible to extract that spectrum from everything else that you get. So in this case, we're measuring the glucose concentration in the bloodstream in a live animal. Okay, now this is some work out of my own research group that is in the area of nanobiophotonics, and it starts with graphene. And graphene, we found a way to make large amounts of graphene in a way that doesn't produce a lot of waste. So typically graphene is grown on copper foils in chemical vapor deposition. You put in some hydrogen gas and some methane, and you heat up the copper foil, and you get a layer of graphene on top of the copper. Then typically what's done is you etch all the copper foil to get you a single layer of graphene. But what we can do instead, and my student came up with this method a couple of years ago, is you take a sacrificial adhesive material and you evaporate it on the graphene film, then you can peel the graphene film off without etching the copper. So it saves all the copper. So this technique, my student actually started a company based on this. It's now in Scripps Ranch. And what you can do then, once you have this graphene on any material you want, is evaporate thin films on it. Okay, so what's the point of doing this? Graphene is so thin that it exhibits a property called wetting transparency. So if you have a graphene film on a surface, the surface energy of the material supporting the graphene penetrates through the graphene and changes its wettability. So if you have a droplet of water and you have some low surface energy things, say you have Teflon below the graphene, the graphene will actually be Teflon-like in its wettability. Then say you have oxidized silicon underneath it, the bead will spread out because the surface energy penetrates through the graphene film because it's so thin. The same thing happens with an evaporated flux of metal atoms. So if you evaporate some metal on a graphene surface that has different backings on it, different metals supporting the graphene, you actually get these different nanoparticle morphologies. So this is gold evaporated onto graphene with copper on the bottom, gold evaporated onto graphene with nickel on the bottom, and you can see that the shapes of the particles go from these sort of largely percolated to largely separated to these crystalline blobs to other types of crystallites. And then if we use silver instead of gold, we can get these blobs of almost spherical particles. So what's cool about these? Well, we know that nanoparticles are active in a couple of different ways. One of which is we can make a strain sensor based on these films. So if you have, say, take this film and you stretch it out a little bit, what happens normally when you apply an electric field across this with some metal contacts is that the current is mediated by hopping from by tunneling from one group of nanoparticles to the next, to the next, to the next. If you stretch it out a little bit, the tunneling probability goes way down, so the electrical resistance goes way up. So you can model this computationally, and we did that. But what you get, what you can do here, this is just a 13 micron step edge, and these are some graphene and metal nanoparticle films. And you press down on here, you can measure changes in electrical resistance that correspond to strains of about 1/1000th of 1%. And you can do some biosensing with this. You can slap it on your wrist, and the sensor is bent here and produces the pulse pressure waveform just by deflection of the graphene nanoparticle films. And you can also make these materials temperature insensitive. So in the case of graphene, we have a material that has an electrical resistance that decreases with temperature, but for metal, the electrical resistance increases with temperature. So if you have something in between, the Goldilocks zone of just the right amount of metal on the graphene, you can actually make a sensor that's not sensitive to temperature at all. And that's this one. So you can do cool things in biomedical nanotechnology where you take these rat cardiomyocytes. None of this is on the exam, it's just for fun. Take these rat cardiomyocytes and they pump. You can see them pumping. So these are rat heart muscle cells from neonatal rats, and they pump on the graphene and they produce this electrical output. Now, why is this interesting? Well, in drug discovery, drug candidates are often thrown out early on in the testing phase because of suspected cardiotoxicity, but there's not really a good way to measure the cardiotoxicity. So perhaps with this method, if you can measure the effect on the ability, or the effect of the drugs on the ability of these cells to pump, then you can tell if it's going to be if the drug is going to be cardiotoxic. Yeah.
I was just wondering, you had the dimension given on the last slide about the sweet spot. What feature is that referring to? Is that interparticle distance for the metal nanoparticles? Or
Oh, it's on this. It's the nominal thickness of the evaporated metal. So if you have an evaporation chamber and you have some amount of, you have what's called a quartz crystal microbalance that measures the amount of metal that's evaporated from your evaporation source, which is just a really hot molten spoon of metal, basically. And the metal atoms impinge on the quartz crystal microbalance, change its vibrational frequency, and then that's converted to mass. And then because we know the density of a metal and the Z ratio, the amount that it becomes thick with a given amount of mass, then we can predict what the nominal thickness is on the actual substrate of interest, not the quartz crystal microbalance. But at low nominal thicknesses of evaporated metal, you always get disconnected islands. Unless you make the substrate in a particular way that allows just small nominal thicknesses to form a contiguous film, that's not usually what happens. Usually you get this balled up, segregated morphology until you reach some threshold thickness where you get deposition everywhere. I know this is a lot, but it's fun. Okay, now optical methods of doing a similar thing. So we know that nanoparticles are sensitive to light and they form these localized surface plasmons. What I didn't tell you is that the gaps between the nanoparticles also affect the surface, the resonant frequencies of these localized surface plasmons. So you have two particles that are very close together, like a couple nanometers. They'll resonate at a different frequency than if they're at, say, 10 nanometers. So you can make a strain sensor that uses an optical signal, because if you stretch these things out, the spacing between them changes. So if the spacing between them changes, the frequency of the scattered light will also change. So in this example, what we did was take these nanoparticles on a flexible glass surface and then bent it. And if you bend a flexible glass surface, the particle separation increases on the top. And so the bent substrate is shown here. And when you bend it, you reduce the intensity of the plasmon resonance between the nanoparticles. And if you have a SIRS active molecule on the nanoparticles, the SIRS intensity goes way down. When you unbend it, it goes back. So the SIRS intensity goes way up here and way down here when you bend it. What if we put cells that contract on them? So in this case, these are myoblast cells, so they're undifferentiated muscle cells. And what we did was get the cells to contract using an electrical signal. So you use the fact that the graphene and the nanoislands are conductive, and you apply an AC signal at this particular signal that induces the cells to contract. So you apply the signal in the gray, and the signal is off in the white regions. Separately, or simultaneously, you take a Raman laser and you measure the Raman scattering intensity. So as the cells contract, they pull the particles apart, the nanoparticles apart, where the cells are adhered to the graphene. So the SIRS signal goes down, just like when we took the substrate and we bent it. In this case, the cells are doing the mechanical work. So we're stimulating the cells to contract, and we're measuring the contractions optically. I do not want to be overwhelming with way too much information in this lecture, so I'm going to take questions or let you all go 5 minutes early. Any questions about this? What I'll say about the exam, we'll do a review on Friday, so everyone's homework, there's no new homework posted this week, but everyone's homework is to come in with a question, and not a question, will such and such be on the test? Let me tell you that the test is multiple choice. It's 100% multiple choice because this has primarily been fact-based as opposed to computation-based. But if you went to the lectures or watched them on YouTube and you know what's in them, you will be totally fine. And I will give away a lot of what's on the test on Friday, so please come. All right, thank you.